In this video, we will continue our discussion about CRISPR and we will learn about what is CRISPR locus. CRISPR locus is basically has all the components that is necessary for CRISPR to function properly. So it is divided into two parts. One is Cas operon, another is Cas array. This is you can consider as DNA sequence, and in this DNA sequence, in Cas operon, it has in Cas operans, before Cas operon, it has this tracer RNA, tracer DNA coding, and in Cas operon, it has four types of or many Cas proteins. Important are this Cas9, Cas1, Cas2. We know the function of these three in the previous videos, that Cas1 and Cas2 helps in cutting the DNA, cutting the foreign DNA that comes into the system for the first time, and Cas9 helps in detecting if the dead DNA again enters the cell and deactivating that DNA. And what this Cas array includes whatever the repeats and whatever the spacers that we get from this function of Cas9, Cas1, Cas2, as we have discussed in previous video, that whatever the genetic information of the foreign DNA is stored as spacer, and each spacer designated as the cell has been infected by this cells has been infected by this DNA by the foreign DNAs that has these elements, and it is separated by the repeats, some repeats. Now we will discuss how this actually functions and make the system immune, or how it is used for gene editing. So in this, so when this is translated, when this tracer is translated, it is called as, it is converted into tracer RNA, and when this Cas9 is translated, it is converted into Cas9, and when this Cas array is translated, it is called pre-CRRNA or pre-CRISPR RNA. What happens is that this is pre-CRISPR RNA. So Cas9 can detect this spacer sequence. It will bind to the spacer sequence, and this tracer RNA will also attach to the Cas9 with the help of linkage. Will also attach to Cas9, and this whole system, this trans-activated CRISPR RNA, this TREC is known as trans-activated CRISPR RNA. Activating CRISPR RNA, trans-activating CRISPR, this tracer RNA. So this tracer RNA helps in guiding this Cas9 to reach to the foreign DNA site, where it will detect the spacer, where Cas9 will detect the spacer, and Cas9 also has the components that chops off the foreign DNA, so that it will not be able to transcribe or translate, however, there is one problem in this, is that this kind of Cas9 system can also detect our own DNA, cell's own DNA, and cut off the cell's own DNA. However, this thing does not happen because Cas9 has additional secondary detection feature, that is PEM sequence. So whatever the spacers that are cut by this Cas1, Cas2 are based on the PEM sequence. Each spacer, after 2 to 3 nucleotides of each spacer, there is some common sequence present in all the foreign DNA that are being converted, that are being stored here as spacer, and these are called as PEM sequence. So how this Cas9 and tracer RNA detects whether the DNA it is attacking is its own or it is foreign DNA. So what it will does is it will detect the spacer sequence, and after that it will also check if there is a PAM sequence available. It will detect the spacer sequence and also check whether there is a PAM sequence available after this after 2 to 3 nucleotides of this spacer, and if the PAM sequence is available, then it gives two-way authentication that this DNA is a foreign DNA. First of all, the spacer is matched, and second, the PAM sequence is also available. So now it can act. This is Cas9. Now it can Cas9 can function to cleave off this foreign DNA. And this PAM is known as photospacer adjacent motif. So in genetic gene editing, what the researchers do is that they will first, if they want to edit a specific if they want to edit a specific protein, they need to identify its sequence and see if the PAM sequence is available or not. And based on the PAM sequence available, as different CRISPR associated proteins has different PAM sequence, and the PAM sequence that is available in the based on the Cas system that they are using. The Cas9 is only for a specific kind of bacteria, but there are now scientists have identified different Cas systems that have different PAM sequence. So what the researchers done is they modified their if there is no PAM sequence present in their protein, they modified their protein with the PAM sequence, and then transfer it with this Cas9 system. So in researchers also what they have done is instead of in bacteria these things are present separately. However, researchers has combined this together and call it as guide RNA. So they can inject the guide RNA, transfect the guide RNA to the modified protein or the protein that have the PAM sequence of the Cas protein that they are using. So in this way, this Cas protein can act on the protein or Cas protein of the CRISPR can act on the proteins of interest or the gene of interest that need to be edited. So this PAM sequence gives additional, you can say that PAM sequence gives additional precision in the gene editing, because if there is no PAM sequence available in the protein, then this Cas system will not function and will not cleave off the sites or edit or do the editing. It does not cleaves off the site of the protein. Okay, and in order to confirm this PAM sequence, researchers has also done like knock out this PAM sequence and try to see the effect, and they have found out that without PAM sequence, the CRISPR is not able to cleave off. This Cas proteins is not able to cleave off the foreign DNA, that modified foreign DNA. So, however, still the detailed function of this PAM sequence is still under investigation and needs further studies. That's all for this video. Thank you.